Hi there class, my name is Danae Hall. Today is September 11th and right now I'm working on the Aseptic Techniques lab. I've got my 2 cups, my gloves, I have extra pairs on the other side, test tube holder, a bottle of bleach. This is my epidermis tablet and the E. coli tablet. I have the pipette, the inoculation loops, a pen, candle, lighter, agar plates that I made last night. So we're ready to go. I washed my hands, workstation is ready and wiped down with bleach. I've got my gloves on, my goggles, and my apron. Alright, so I don't think there's any more alcohol in here. Been squeezing it out a little bit, so this seems pretty sterilized. I'm going to remove the lid of the nutrient broth vial and pass the top of this through the candle. So I just want to make sure that I don't spill any into the candle. And then I'm going to do the same thing with the E. coli. Now I'm going to take 0.25 milliliters of the nutrient broth vial and then pipette it into the culture vial. So it's a 0.25 and I'm just going to pipette it in. This is the culture vial that I'm shaking until the tablet dissolves. Pipette the culture into the nutrient broth tube. I'm just going to pass the lid of this through the candle one more time to sterilize it. We're going to do the same thing we just did with the E. coli one with a new one, and we are going to label this one S. epidermis. So it says S. epidermis tablets. Pass the lip of each one of these tubes through this flame. Alright, so the next step is to take 0.25 milliliters of the epidermis nutrient broth and place it in to the epidermis culture vial and shake it until the tablet is completely dissolved in the nutrient broth. Pipette the culture back into the nutrient broth vial. And then put the cap back on. Alright, so I have about a 1/4 cup of water with about a 1/4 teaspoon of yeast. Alright, so I am working on my last one because the yeast has been sitting for over 10 minutes now, so it's just about time to start. I'm going to take the S. cerevisiae tube. I'm going to pass this through the candle. 0.25 milliliters of the yeast. Pipette that into the S. cerevisiae. Lip through the flame one more time. E. coli, epidermis, and the S. cerevisiae. Take my 3 culture vials and then put them in the place that I decided I would have be my incubation spot, and I will keep them there for about 48 hours. So I left the cultures incubate for about 48 hours and there wasn't a ton growing in them, so I left them in there for an additional 24 hours. So I have 3 plates ready to go. One of them is larger because I had one break in the box and they haven't sent me a new one yet. So just using that one. Candle, lighter, 3 inoculation loops. They're in the alcohol right now. And then I have my 3 cultures, a pen, paper towel, my mask, some bleach, and some alcohol. The plates are all now labeled with their corresponding culture name. So right now I am just shaking dry the inoculation loop, and then I'm going to streak the third quadrant of the S Cerevisiae plate, put the lid back on, and place the inoculation loop back in the alcohol for 20 seconds before shaking it dry and doing the fourth quadrant. I'm on my last tube right now. This is S. epidermis. After 20 seconds, I'm going to take the lid off, shake the inoculation loop completely dry, and then I will come back to my epidermis plate and streak the second quadrant. And I will do this until all 4 quadrants have been streaked. Alright, so that is the end of that part. I'm going to take the 3 new plates and incubate them for about 48 hours. Here are the 3 colonies that have been incubating for 48 hours. So this is S. epidermis right here. E. coli is right there. And S. cerevisiae is right there. So you can kind of see the streak marks going on each quadrant.